Behavioral Neuroscience, Chapter 17, Segment 3. A variety of brain regions are involved in different attributes of working memory. For testing spatial location memory, the 8-arm radial maze was used. In this maze, rats must recognize and enter an arm of the maze that they have been down shortly beforehand. The rats were tested following surgical lesions of the hippocampus, the caudate nucleus, and the extrait cortex. The animals with the hippocampal lesions were impaired. For testing memory of motor behavior, rats must use working memory to remember whether it made a left or a right turn a few moments before, and they receive an award if it makes the turn in the same direction in the follow-up trial. The only animals with lesions of the caudate nucleus were significantly impaired. For testing working memory in the sensory attributes, the rats were required to identify the novel stimulus in each pair of stimuli presented. Only rats with extrait lesions were impaired. The brain regions that do not do the initial processing of stimuli often act as memory buffers for holding up a stimuli in working memory. Delayed response tasks work by varying the delay time between presentation and removal of a stimulus and the response. Brain regions involved in learning and memory. An interim summary. Many regions of the brain are involved in learning and memory. Different forms of memory rely on at least partially different brain mechanisms, which include several different regions of the brain. The same brain structure can be a part of the circuitry for several different forms of learning. Neural mechanisms of memory storage. Neuroplasticity is the ability of neurons and neural circuits to be remodeled by events. It is found in all animals and indicates that it is an ancient and vital product of evolution. Memory storage requires neural remodeling. Charles Sherrington speculated that synaptic alterations may be the basis of learning. Plastic changes at synapses can be psychological or structural. Synapses changes that may store information can be measured physiologically. They should be presynaptic, postsynaptic, or both. Changes include greater release of neurotransmitters, molecules, and/or greater effects because the receptor molecules became more numerous or more sensitive. The result of change is an increase in the size of the postsynaptic potential. Synaptic activity could be modulated by inputs from other neurons, causing extra depolarization or hyperpolarization of the axon terminals and changes in the amount of neurotransmitter released. New synapses could form or be eliminated as a function of training. Training can lead to the reorganization of synaptic connections. Varied experiences in learning can cause the brain to change and grow. In standard studies of environmental enrichment, rats are randomly assigned to 3 housing conditions. 1. Standard condition. Animals are housed in small groups in the standard lab cages. 2. Impoverished condition. Animals are housed individually in standard lab cages. And 3. Enriched conditions. Animals are housed in large social groups in special cages containing various toys and other interesting features. Compared to the IC animals, EC animals have heavier, thicker cortexes with enhanced cholinergic activity. Cortical neurons in the EC animals also have more dendritic branches, especially on dendrites closer to the cell body than those from the IC animals. It is difficult to find the exact synaptic changes that underlie a particular instance of learning because there are so many synapses. Invertebrate nervous systems, slow plasticity. The sea slug aphasia is capable of non-associative learning. Non-associative learning is a type of learning in which presentation of a particular stimulus alters the strength and probability of a response in according to the strength and temporal spacing of that stimulus. There are 3 types: habituation, dishabituation, and sensitization. Habituation is a decrease in response to a stimulus as the stimulus is repeated. Dishabituation is the increase in response amplitude. Sensitization is the response is greater than the baseline level because of the prior situation. Synaptic plasticity can be measured in simple hippocampal circuits. Donald Hube proposed that when the presynaptic neuron repeatedly activates a postsynaptic neuron, the synaptic connection between them will become stronger and more stable. Cell assemblies are large groups of cells that tend to be active at the same time because they have been activated simultaneously. Hebron synapsis is a synapse that is strengthened when it successfully drives the postsynaptic cell. Cell assemblies are linked via synchronized activity of the Hebron synapsis and then can act together to store memory traces. Tenses are an intense volley of the action potentials, and long-term potentiation is a stable and enduring increase in the effectiveness of synapses following repeated strong stimulation. It can be generated in conscious and freely behaving animals, in anesthetized animals, and in tissue slices. LTP occurs at several sites in the hippocampal formation. The hippocampal formation consists of two interlocking C-shaped structures, the hippocampus and the dene gyrus along the adjacent cortex. Neuroscientists distinguish three major divisions within the hippocampus labeled CA1, CA2, CA3. LPT was originally demonstrated in the main input pathway to the hippocampal formation. In CA1, LTP occurs at synapses that use excitatory neurotransmitter glutamate. It is dependent on glutamate receptor subtype NMDA receptor. Treatment with drugs that selectively block the NMDA receptor completely prevent the new LTP in the CA1 region, but it does not affect the already established LTPs. NMDA receptors and AMPA receptors collaborate in the LTP. NMDA receptors cannot respond to the glutamate because magnesium ions block the NMDA receptors integral calcium channel. AMPA receptors emit sodium ions when activated. The increased activation of the AMPA receptors depolarizes the postsynaptic membrane. A large influx of calcium at the NMDA receptors activates the intercellular enzymes called protein kinesis that alter or activate a variety of other proteins. One of them is called CAMK2. The activated CAMK2 causes more AMPA receptors to be produced and inserted into the postsynaptic membrane and existing nearby AMPA receptors, which are induced to move into the active synapse. They are modified to increase their conductions of sodium and potassium ions. CREB is the transcription factor, a protein that binds the promoter region of genes and causes those genes to change their rate of expression. That is activated by protein kinesis, including CAMK2. The direct result of activations of the NMDA receptor in the activations of CREB and changes in the expression of genes encoding a wide range of proteins. When the postsynaptic cell is strongly stimulated, its NMDA receptors become active and emit calcium. An intracellular process causes the postsynaptic cell to release a retrograde messenger that travels back across the synapse and alters the functioning of the presynaptic neuron. This ensures more glutamate will be released into the synapse than previously. LTP can occur without NMDA receptor activity, and some forms are blocked by drugs with completely different modes of action, such as opiate antagonists.